The first event of skeletal muscle contraction is the muscle fiber has to get excited by a motor neuron. So this occurs at the neuromuscular junction and results in acetylcholine, the neurotransmitter, being released from the synaptic knob, and then it binds to the acetylcholine receptors on the surface of the sarcolemma. So a nerve signal, also called a nerve impulse, is the electrical signal that propagates down an axon of a motor neuron. Now, the nerve signal is going to trigger the opening of voltage-gated calcium channels down here on the synaptic knob, and then calcium will move from the interstitial fluid into the synaptic knob. Well, that is going to cause the vesicles to release acetylcholine. So the binding of the calcium to those synaptic vessels triggers the merging of the vesicles with the membrane part of the synaptic knob. This results in exocytosis of the calcium from those vesicles into the synaptic cleft. Remember, that's the space between the nerve cell and the muscle cell. Now, acetylcholine is released from approximately 300 of these little vesicles per nerve signal, but each of those vesicles contains thousands of molecules of acetylcholine. Since we're talking about neurotransmitter acetylcholine, let's look at a disorder associated with that, and that is myasthenia gravis. So asthenia means weakness. So this is an autoimmune disease. It affects about 1 in every 10,000 people, but mostly women, and mostly women between the ages of 20 and 40. Because it's autoimmune, that means a person's immune system is creating weapons called antibodies against their own cells. So these antibodies will attach to some of the structures in the neuromuscular junction. When it does so, it causes those acetylcholine receptors to group into clusters abnormally. Then the immune system is going to break down all of those clustered up receptors. Now, that means the muscle cell fiber is losing a lot of acetylcholine receptors, and the importance of that is you've got to have acetylcholine receptors for acetylcholine to bind to. So what is that going to do? Well, it's going to result in decreased muscle stimulation, rapid fatigue, and muscle weakness. The eyes and the facial muscles are usually the first that show the signs of this, and it can produce double vision and drooping eyelids. Afterward, it's usually followed by problems with swallowing, weakness in the limbs, and just a decrease in stamina. Now, depending on the severity of this, some patients will live a normal life with a normal lifespan. Others will die within a very short time if the respiratory muscles are affected and paralyzed, and thus breathing cannot occur normally. The second event of muscle contraction is excitation-contraction coupling. So this involves the sarcolemma, the muscle cell membrane, T-tubules, and the sarcoplasmic reticulum. So excitation coupling couples the events of skeletal muscle contraction, excitation, at the neuromuscular junction, and then leading to contraction caused by sliding of the myofilaments in the sarcomeres. Okay? So these events are coupled in what we call excitation-contraction coupling, or how excitation leads to contraction. So quick overview again. Here's the axon terminal. That's the same thing as the synaptic knob. It releases acetylcholine into the synaptic cleft, the space. It binds to acetylcholine receptors. The sodium gates open, sodium rushes in and depolarizes this part of the membrane, which depolarizes the next and the next and the next as more and more sodium gates open. The T-tubule depolarizes because it is also cell membrane. This causes the sarcoplasmic reticulum to dump out calcium. The calcium then binds to the troponin. The tropomyosin moves off the binding sites. Now, myosin heads can bind to the actin, and when they bind together, the resulting action leads to contraction. So those acetylcholine receptors are chemically gated ion channels that are stimulated to open just for a short period of time when acetylcholine binds to them. The opening of these channels allows sodium to rapidly diffuse into the skeletal muscle cell and potassium to slowly diffuse out. Now remember, both of those are positively charged. So if more sodium is coming in than potassium is going out, the inside of the cell is becoming more positive instead of more negative. So this results in a gain of positive charges on the inside of the skeletal muscle. When you talk as much as I do, sometimes the words just don't come out right. So these changes in the membrane potential in the motor end plate are very short-lived and localized, but if there's enough gain of positive charge to change the resting membrane potential from about negative 90 millivolts to negative 65 millivolts, then an end plate potential is produced. So think about that. If we're measuring the voltage inside this cell, right beside the cell membrane, it should be approximately negative 90 millivolts. But for every positive sodium that comes in, it basically takes away one negative charge, because positives and negatives sort of cancel out each other, if you will, right? So the more sodium comes in, the less negative inside that cell membrane becomes. That is what we call the resting or the end plate potential, and it's the minimum change in voltage or the threshold that must be reached that will trigger the opening of the voltage gated channels in the sarcolemma to initiate an action potential. Guys, I'm sorry, that just went crazy there for a moment. Let's go back over that. So when we're bringing those positively charged sodium ions in, the inside of the cell is becoming less negative. So when that resting membrane potential goes from about minus 90 millivolts to about minus 65 millivolts, and end plate potential is produced. So the end plate, of course, is just this area right here of that muscle cell membrane. So an end plate potential is the minimum voltage change in the motor end plate that will trigger the opening of voltage-gated channels to initiate an action potential. Now remember, action potential leads to action. What's the action of a muscle cell? To contract. The end plate potential triggers an action potential that's propagated along the muscle cell membrane and the T-tubules, because remember, they're just part of the muscle cell membrane. Now, I really like this image because you can see here with the bright yellow, it looks like it's lit up. This is your sarcolemma, and it extends down into the cytoplasm of the cell and circles around the myofibrils there, okay? So what that is doing is bringing parts of the cell membrane down into the cell instead of just forming that outer covering, okay? So everything you see here in yellow is cell membrane. We call the outside part the sarcolemma. We call this inside part the T-tubules or transverse tubules because they transverse the cytoplasm. Okay, now with that said, an action potential involves two major events. Depolarization, which causes the inside of the muscle cell to become more positive due to that influx of sodium. And if we depolarize it, yep, we got to repolarize it. So that is simply returning the inside of that cell membrane to its relatively negative resting membrane potential. because we're pumping out those potassium ions. So the electrical change of the end plate potential at the motor end plate stimulates the opening of voltage-gated sodium channels in the adjacent area of the cell membrane. So think of it this way again. Go back to the dominoes. If one domino falls and hits another, and another, and another, it's propagating along that chain of dominoes, because one caused another to do something, caused another to do something. Well, what's happening with this is when the sodium gates open, that positively charged sodium comes in, that part of the membrane depolarizes, and that triggers the very next set of sodium gates in the membrane to open, because they're voltage gated. The change in voltage of the membrane causes them to open. So that's one domino causing another domino to fall. Okay. Now, as this is happening, the internal part of the sarcolemma was relatively negative, but it becomes relatively a little more positive with a change in the membrane potential from the threshold that we mentioned a while ago. Remember, we started out with resting membrane potential at -90. Threshold was about -65 millivolts. But if we bring in enough sodium, that membrane is going to go all the way to +30. So that reverses the polarity of the cell membrane, and that is what depolarization is. Now, depolarization is going to continue until we close those sodium gates. All right, just to reinforce that, okay? The propagation of depolarization along the muscle cell membrane and the T-tubules requires the sequential opening of voltage-gated sodium channels. The inflow of sodium at that first place, at that motor end plate of that sarcolemma, causes the adjacent regions of the sarcolemma to depolarize also by opening those sodium gates. Now, that is called propagating an action potential along the sarcolemma and the T-tubule. Again, think of the dominoes. So let's look right here. So the ligand would be acetylcholine. If it binds that receptor on the sarcolemma, it causes the gate to open, and positively charged sodium comes from the outside of the cell to the inside. Now look over here, we've got lots of negative charges, so this is cell at rest. That is resting cell membrane potential, okay? Now that positively charged sodium rushes in, and look, now we've got positive charges around the cell membrane, and if it reaches -65 instead of -90, then it has reached threshold, and it propagates that action potential all the way down the sarcolemma and the T-tubules. Now, potassium gates are going to open with a little bit of potassium going out to help restore that negative inside the cell. This is known as repolarization. So for every sodium that goes, we do 3 sodiums one way, 2 potassiums the other way. Okay, so that's how we reset that membrane potential. Pump 3 sodiums out, bring 2 potassiums back in. Repolarization is necessary to reestablish the resting membrane potential, and that allows that muscle cell to respond again the next time it's stimulated by acetylcholine to propagate a new action potential. Now, the way I think of this is to think about what is going to happen. It leads to this refractory period. So when your cell is depolarizing or repolarizing, it's in what is called a refractory period. That is very important because that's a brief period of time where the muscle cell can't be re-stimulated. So in order for a new action potential to occur, we got to reestablish the resting membrane potential. So I think of this, and I use this example in my class a lot, of flushing a toilet. If you think about it, when you flush the toilet, the water is leaving the tank and coming into the bowl, and then the tank has to refill before you can flush it again. Excuse me. So try it. You flush. Reach around there and try to flush again immediately. Well, that's not going to work. You've got to allow it to reset. Well, that's exactly what has to happen to these muscle cell membranes. If they contract because they depolarized, the only way they can ever contract again is to repolarize, because remember, depolarization led to contraction. So let's look at the summary slide now for excitation-contraction coupling. So the acetylcholine is released from the synaptic knob or the terminal knob, whatever you prefer to call it. I don't care, just know how it works. So the acetylcholine is released via exocytosis into the synaptic cleft, where it binds to receptors on the motor end plate, that little part right there of the muscle cell membrane, the sarcolemma. When it binds to the receptors, the sodium gates open and sodium rushes into the cell. Sodium is positively charged, so it negates the negative charges right here at the cell membrane inside the cell. So when enough sodium diffuses in to change the resting membrane potential from -90 to -65, that results in an end plate potential. Okay, now let's move on over here. The end plate potential initiates an action potential, depolarization followed by repolarization. So depolarization is when the voltage-gated sodium channels open, that positively charged sodium moves in, and when enough positive charges come in to cause that membrane to go from -65 threshold to +30, that cell membrane is no longer polar, it is depolarized. Remember, polar simply means different on one side versus the other. Okay? Now, we have to repolarize this cell for it to react or do its job again. So voltage-gated potassium channels open after the sodium channels. That starts pushing potassium out. So it's slowly bringing the inside of the cell back to more negative. But now we've also got to start pumping out more sodium. So we pump out more sodium, pump in more potassium. This restores those positive charge or the negative charges back in the cell membrane, because we're getting rid of more sodium than we're bringing back in potassium. So it gets the membrane back to resting membrane potential of -90 millivolts, and that cell is now repolarized, or if you go back to my analogy, you can flush the toilet again.